Thanks, Ned. I'm going to go ahead and share my screen, and hopefully everyone will be able to see what I am presenting. Does that look good?
Looks good.
Okay, yeah, so I just thought I would take this opportunity today to provide an update on a pilot study that we conducted in the Hudson River Estuary looking at carbon dynamics. And before I get started, I just want to acknowledge some people who were really helpful to getting this work done. First of all, Jay Siston really helped a lot with the logistics of our mooring deployments. William Pardis and Shawnee Trailer were really helpful in our along estuary surveys, where we used a gas analyzer that was developed by a colleague of mine at Woods Hole, whose name is Anna Michelle. So it's her technology that we're using. And I also want to thank Ned for getting this group together and the leadership that he's provided. And that's really led to some funding opportunities for me. And specifically, I'd like to thank the Krupp Foundation for their financial support for the research that I'm going to present, along with an anonymous donor who's a friend of both Woods Hole and Scenic Hudson. And so their funding, which really I don't think would have happened without my participation in this group, led to what I'm going to show you today. So before I get started, I'm going to start with a basic cartoon that I've showed a couple of times in these meetings that we've had together, and it really presents my view of carbon, and as Ned said, from sort of mountaintop to ocean floor. And so a lot of what we're talking about deals with regenerative farming practices and the role that they can play in pulling in atmospheric CO2 and sequestering that carbon in the soils. But I think humans are altering the carbon cycle in a lot of different ways. And while agriculture is certainly a powerful way of helping to mitigate some of the anthropogenic effects of this alteration of the carbon system, there are a lot of other things going on as well. And I think a lot of people in this group have interest in these areas. And so when we think about the watershed, there's forest management practices, and those certainly play a role in how much carbon is getting into the local watersheds. So that is another source of carbon. We have wetlands and marshes along the system, and they pull in a lot of atmospheric CO2, but they're also thought to be big sources of organic carbon into riverine and estuarine environments. And so that organic carbon is another source that could potentially fuel CO2 emissions as this carbon moves down river and into the estuaries. There's a lot of industrial practices that have impacts on both CO2 and methane. And I'm going to be talking very briefly today about sewage treatment, which I think in the Hudson River plays a big role. And so these sewage treatment plants, they're putting organic material into the Hudson River, and they also, we think, are a big source of methane. And I'll touch briefly on that today. And so, humans are impacting the system from the mountaintop all the way to the ocean floor and all along the way. And my interest is really looking at the estuary, which is the sort of region where the riverine waters meet the ocean waters. And that's an important region because that's where a lot of the organic carbon that's being input into the watershed is ultimately received and processed before it makes its way either back into the ocean or into the atmosphere or getting buried. And so not only is carbon entering these systems, but nutrients from farms and other sources is entering as well. And that plays a big role in governing things like the algal production. We call them phytoplankton, but essentially they're the plants of the ocean, and they respond very strongly to inputs of nutrients. And when they die, they sink to the bottom, and they can lead to low oxygen conditions. And those low oxygen conditions have big impacts on water quality, as well as the pH of the water, which ultimately determines how much CO2 those waters can carry. And so these estuarine zones are essentially where the river and the ocean mix. And so we're pulling in ocean water. That ocean water has carbon in it. A lot of times that's inorganic carbon, but there's organic carbon in those ocean waters as well. And they can get pulled in, and they all get processed in this estuarine environment. And my interests really are in these estuarine environments that exist in the sort of the interface between the terrestrial and the marine environments. And what I'm showing here on the left is a map of the Hudson River watershed. And this is really this huge area where we have forests, we have farms, we have urban environments, and they're all putting carbon and nutrients into the riverine waters. These riverine waters transport that material towards the ocean. And in a simple sense, you can think of the river as being a very simple hose, and it's delivering material down towards the ocean. And it's going to continue to move downstream until it reaches the estuary. And the estuary is where the sort of circulation patterns really change. This is a region where we're pulling in ocean water along the bottom, we're mixing it with the river water, and we're outputting brackish water. And so these estuarine environments tend to be extremely effective at trapping material. That's where we trap a lot of organic material. And as a result, we have very, very high carbon fluxes in these environments. And so this is where a lot of the carbon gets processed. And so that's really where my interests come in. I'm studying the physics of these systems, but I'm also very interested in how the physics impacts the biogeochemical cycling. And particularly recently, I've gotten very interested in how estuaries process carbon and how ultimately that affects how they emit CO2 to the atmosphere. And I think if we really want to understand human impacts on carbon cycling, estuaries are a key environment to make some of these measurements and study these processes. So as I said, I was very fortunate to get some funding from the Crump Foundation and some others to do a pilot study to look at some of these dynamics. And so I just want to walk through that study really quickly and present some of the preliminary results. This was something we conducted from August 10th to October 14th, this past late summer and early fall. And as part of that study, we deployed some instruments, and that's what I'm showing on this slide here. So on the left, we deployed a mooring with a surface buoy, and attached to the surface buoy and on a wire going down from that surface buoy, we had a series of instruments that measured the temperature, the salinity, and the overall dissolved oxygen in the water column. We call those instruments CTDs. And then adjacent to this mooring, we deployed a bottom tripod. This bottom tripod sat on the bottom, and it made measurements of the currents. So we had something called an ADCP current profiler. So that's making vertical measurements of the currents throughout the water column. We had another CTD on that frame as well, measuring salinity, temperature, and dissolved oxygen. And then one of the things that was unique about this study is we had a sensor that was measuring the concentration of dissolved carbon dioxide in the water column, this PCO2 sensor. And so we were able to continuously measure the amount of carbon dioxide in the water column for about a 2-month period this summer. So this is what the tripod looked like when it went in. This is what it looked like when we pulled it out. So it's covered with barnacles. This is our PCO2 sensor. So again, these are challenging measurements to make because of all this organic productivity. But all this organic productivity is why there's such a huge signal when we start looking at things like carbon dioxide and oxygen dynamics in these systems. We deployed those sensors from a 37-foot research vessel called the Panaway, and we deployed that gear right off of Croton Point. And so just to give you some orientation, this is Haverstraw Bay that I'm showing. The Tappan Zee Bridge is to the south, and the Bear Mountain Bridge is to the north. And during this period of time, the salinity from the ocean had extended all the way north of the Bear Mountain Bridge. And so that riverine water is coming down, is meeting the ocean water, and it's mixing essentially anywhere south of this region. And so this is really a key transition from the riverine to the oceanic waters. Another key component of what we did is we did along estuary surveys. We went all the way from the tip of Manhattan up to West Point. That's a distance of, say, 80 to 90 kilometers. We did that in a 25-foot Parker, which is a small vessel that we chartered out of Haverstraw. And the key equipment that we used was my colleague Anna Michelle's gas analyzer. That's these yellow Pelican cases that you see on the left in the cabin of this small boat. And that gas analyzer was attached to a centrifugal pump that had a hose. And so essentially what we did is we pulled water up from various depths and ran it through this gas analyzer to determine the concentration of CO2 and methane in the water. So the end of this hose was attached to a CTD profiler. So that's a device that measures temperature, salinity, as well as dissolved oxygen, turbidity, and then chlorophyll fluorescence, which is really how much plant material is, living plant material is in the water column. And so a lot of people have made measurements of surface PCO2, but to my knowledge, no one has really mapped the full three-dimensional distribution of CO2 in the water column. And so these are sort of the first measurements of that kind in an estuary. So I just want to jump into some of the data and give you a quick taste of what we measured. So this is an overview of those in situ instruments that were deployed at the bottom in the mooring. So the x-axis is just time going from early August to sort of mid-September that we deployed the gear. This upper panel is just a measure of the tidal range. And so the tides in the Hudson River vary in relation to the moon and the sun's relationship to each other. So we have periods where we have big tides approaching a tidal range of say 1 meter. And then we have periods with much weaker tides where the tidal range is say 0.5 meter. And that mixing by the tides plays a really big role in what goes on in the water and how the salinity, how the ocean waters and the riverine waters mix. So this second panel shows the top to bottom difference in salinity. I call it salinity stratification, but you can think of that simply as what's the difference between the salinity of the surface water and the salinity of the bottom waters. The surface waters tend to be fresher, the bottom waters tend to be saltier, but that's very dynamic and changes with inputs from the rivers as well as the tidal mixing. And what you can see in this plot is that sometimes there's a very big difference in salinity. The surface waters are about 10 practical salinity units lower than the bottom waters, and that tends to occur when there's very weak tidal mixing. When the tides are strong, the water column gets mixed up, and that difference in salinity from the surface to the bottom gets essentially erased. And that has really big implications for the gases in the water column. So when we have these periods of very strong top to bottom salinity differences, the bottom waters are effectively cut off from the atmosphere. And so any organic carbon that's being respired or consumed is going to draw down the oxygen. And so you can see from this bottom dissolved oxygen sensor, during these periods of strong vertical density stratification, the oxygen will begin approaching 0. And that has significant implications for the organisms that live there. Now, at the same time that the oxygen is being drawn down, that respiration of organic carbon is building up the PCO2. And so the dissolved gas carbon dioxide in the water column is getting to be, has very high values when the mixing is weak, the stratification is strong, and the oxygen is low. Now, as we go a couple days later, maybe a week later, we see very dramatic decreases in the bottom CO2. So we're going from say 4,000 down to 2,000. So these are extremely high values. The atmosphere is something like 400. And so these are sort of a factor of 10 bigger than atmospheric values. And so the tides are effectively mixing up the water column, and that plays a really big role in how much of these gases can exchange with the atmosphere. And to illustrate that a little more clearly, I'm going to show the data from our surveys. So to orient you, the x-axis is simply degrees north, or the distance north up the system. And so the left-hand part of the panel of these plots is the tip of Manhattan, and the right-hand side is West Point. So the ocean in these plots is to the left, the river is to the right, and the y-axis is depth. These black lines are the salinity of the water, how much salt is in the water. And what you can see is we have these black lines, so there's essentially 0 salt if we go far enough to the right, and there's high salinity to the left. These black lines are the contours of salinity, and one of the things we'll note is that particularly on this August 12th survey, the surface waters are very fresh and the bottom waters are very salty. And as a result, we can see what effect that has on both oxygen in the upper panel and CO2 in the bottom panel. So the oxygen is measured in terms of percent saturation. So if the water was just in equilibrium with the atmosphere, everything would be at 100%, which would be sort of a teal green color. But what we see on this August 12th date is that the surface waters were very, very super saturated in oxygen. So they had much more oxygen than they normally would have had. And these bottom waters were very low in oxygen. So they have much less oxygen than they normally would have. That corresponds to the CO2, which in these blue values in the lower panel, that's very low CO2, and the red is very high CO2. And so the water column structure with that big gradient in salinity is decoupling the bottom waters from the atmosphere, and we're drawing down the oxygen to very low values, and we're building up very, very high values of CO2. What's driving some of this is the fact that there are plants living in the water. So the next thing I'm going to superimpose over top of this is the chlorophyll fluorescence. So this is a measure of how much algae is in the water. And we see that there's this red stripe near the surface. These are plants that live in the water. And so what they're doing is they're producing oxygen, creating that supersaturated surface condition of oxygen, and they're drawing down the CO2. Now, ultimately, that plant material is going to sink to the bottom. It's going to add more organic material that's going to be respired that's going to help draw the bottom oxygen down. But as a result of this bloom of algae in the sort of mid to lower estuary, we have very low surface values of CO2. And as a result, a lot of the estuary is pulling in carbon. There's an undersaturated value of CO2, so it's pulling in carbon in the lower estuary, but in the upper estuary, where it's well mixed, it's outgassing a lot of carbon. And if you averaged over the whole surface area that we surveyed, there would be a net export of carbon, or an emission of carbon, of about 95 grams of carbon per meter squared. Now, if we go simply 10 days later, during a period of strong tidal mixing, we see that everything's now been mixed up. That oxygen that was strongly segregated in the vertical is now much more uniform. It's slightly undersaturated everywhere, but the patterns are much weaker because the tides have essentially mixed everything up. And the same is true for the CO2. All that really high CO2 in the bottom waters has now been mixed up to the surface. And when it gets mixed up to the surface, it can now freely exchange with the atmosphere. And so all of that really high CO2 has now been mixed up to the surface, and now the entire estuary is outgassing from the ocean to the river. And so we have the chlorophyll fluorescence in this upper panel. That bloom is now gone. It's been mixed away. There's no longer a very thin layer of algae at the surface. And we have CO2 emissions that are outward everywhere along the system, and they've increased by about a factor of 4 to 5. So we have this sort of breathing of the estuary that's being modulated by the tides. And so all of this organic carbon is being produced, it's being consumed, and that's building up the bottom waters in high values of CO2 that can't really outgas until they get mixed to the surface. And again, these are sort of the first really comprehensive measurements of the three-dimensional distribution of CO2 that have been made in an environment like this. And so I just want to end with one final plot comparing the distribution of methane. This is data that we just got this week, actually, in terms of getting the calibrations correct. And it's sort of hot off the presses, so I don't want to spend too much time discussing it. But these are two plots comparing the two cruises on August 12th and August 22nd. Again, the x-axis is the distance along the system with the ocean to the left, the river to the right. And this is, the colors represent the methane concentration. And so I just want to call your attention to a couple of key features. On August 12th, there was this hot spot of methane at the bottom waters, sort of midway up the estuary. And this is immediately adjacent to the North Point sewage treatment plant. And so what we think is that the methane is actually a really good indicator of inputs from the wastewater treatment plants. When we went back on August 22nd, we see that there's high methane, much higher methane in the lower estuary. And we think this is either water that has come around the tip of Manhattan from the East River, or that this is actually water from New York Harbor. But either way, it's got very, very high methane. And methane is another really important greenhouse gas that humans play a big role in modulating. And so we think the methane is actually a good tracer of wastewater and sewage inputs from the sewage treatment plants that line the Hudson River. So with that, I'll just come back to my sort of overall cartoon about carbon without borders, and this need to sort of understand all of these various components of carbon cycling to really get a good handle on how humans are altering anthropogenic carbon emissions. And again, I really think a group like this is a unique group in a way that has expertise in all of these various these areas. And so I've been really fortunate to be a part of this group. And without my participation, I don't think the research that I just presented would have occurred. And it's really been beneficial for my research specifically. And so I'll go ahead and end there, and I'm happy to take any questions. And I apologize for the technical difficulties.